for downstream analysis. And your downstream analysis can either be for RNA, DNA, or protein analysis. Usually, the slides that you use for this type of microdissection requires a membrane. So you could have a slide like this, an ordinary glass slide, where you have a very thin strip of membrane. This membrane, in this case, is polyethylene naphthalate. It's a UV absorbing membrane because what we use for cutting tissue is a UV laser, 355 nanometers. So that laser cuts from beneath. So you have your glass slide and then your membrane is on top. And then on top of the membrane is the tissue that you want to isolate. So your membrane cuts from below here. It cuts through, penetrates through the glass and cuts the membrane along with the tissue, and then lifts the tissue with one pulse. It lifts it into a collecting vessel. So you have your collecting vessel put right above the tissue, and that's where it collects. Your collection can either be, with a buffer in a cap or a dry type of collection. So we have this microcentrifuge tube. In this case, it's an adhesive type of cap. So you can have two different kinds of caps actually. One is a cap with an opaque adhesive on it, and the other cap has a clear adhesive. So there's a clear adhesive here. The only difference between the two is, well, this one here, the opaque one, would give you more contrast if you have a stained tissue. Okay, so anything that's stained will be easily seen with this type of cap. Whereas if you have a clear adhesive cap, then this would be normally used for very small parts of your sample, maybe like chromosomes, or if you have unstained tissue, this would be ideal. Okay? So it's just basically for viewing purposes, because in this system you have the ability to view whatever you have cut right after. Okay? So the software we use is called PAM Robo there. So what you need to do is just double click on the software, and then it starts initializing. When it initializes, the stage would move. Okay, it's just sort of like a test, testing all the components. So this stage is movable. The objectives go down, focus up and down. You have a cap mover here. This one is for putting in your collection device. So depending on what you have in your lab, so we have collectors where we put your adhesive cap on. So you have this type of collector which can hold 2 adhesive caps. Then you have also a single tube one. This one, so you put in one tube, or you have one where you only have the cap. So basically you can cut off the cap from your centrifuge tube and then just collect using the cap. This usually is used if you're collecting live cells. This one. Alright, so once you have your software initialized, you can, go ahead. So you can, it's open actually right now. So we have all these settings here. You can load a setting that you had previously saved, or you can load a default setting. So you go to settings here and you go to load settings. And usually for our users, if it's the first time they're using, they usually load the default settings. If you don't load anything, then actually the default settings are already loaded. Okay, so let's go to users. There are users here. And you go down. So the default we have default settings for each of the objectives, because once you load the setting file, it's only specific for the type of objective that you're using. So let's just use the 5x for now. Okay. So settings are loaded. You have the cameras ready. There are 2 cameras in this system. This is the first camera, and it's an action cam 1cc. It's this one has color, and it's usually used for stained tissue, and that's what we're gonna use. There is another camera over here at the sideboard. It's a camera which is actually just monochromatic, but it's used for fluorescence. So this system can be used for stained or unstained tissue, as well as for tissue that has some fluorescent material. Okay. So we have here the different objectives that are in place right now. We have 5x, 20x, and 40x. All of these 3 objectives are air objectives. They have a long working distance, so you could use that to both visualize and use that to, for cutting your tissue. Then we have a 150x objective. This one is usually used for viewing like chromosomes, very small material. It's a glycerin type of objective. It is a very short working distance, so you wouldn't use an ordinary glass slide for this. You'd have to use a very thin, cover slip thick, like a 0.17 millimeter thick membrane slide for this. Okay. It's actually very good also at visualizing those small particles. Although it's a little bit more difficult to cut just because there's a glycerin in there. Okay. So all you have to do to choose your objective would be to click on, whatever is displayed here. We also have this touchscreen monitor, so you can also choose your objective from here. This transmitted light illumination would be used for your unstained tissue as well as for stained tissue. So basically you have just a halogen lamp which goes down through this condenser here, and then the light would be coming out here, and then you could easily visualize your sample. Okay. So right now the light is turned off. Once you initialize the system, it would turn on automatically. You could turn on your light using the touch screen, so you can turn it on from here, or you can just click on this light icon here.
You have fewer laboratories.
Okay, so you'd be able to start visualizing your sample. The amount of light that gets into your sample, or basically your exposure time, can be adjusted by using this auto live mode, so it automatically adjusts your exposure time. Or if you don't like that, you can also just click on this measure here. So it would like calculate, how much time it would take to be able to visualize your sample. You could also manually increase or decrease the exposure time by using these arrows here, or you can just type in a number. Okay. Normally, if you're dealing with stained samples, you don't really need to do a gain, because there's a gain setting here which would usually come out if you're doing fluorescent samples. There's a white balance button here, so if you see maybe some color there, you don't like the color, you can just click on the white balance and the color would be adjusted. Okay. Alright, so if you're doing just a stained tissue, you would normally be under this common tab here under the microscope setting. There's also another tab here, reflected light, and that's usually for fluorescent samples. The settings here, if you click here, okay, you can just click on the microscope here, and then you can assign certain settings as to,
So basically,
where your light is going to go. If you want your light to go just on the eyepieces, or just the camera, or you can split them out.
So the last person who used it probably
So these settings you can like set it up once you set up your microscope. Okay, so you can just exit that
red ones can be weird.
and go back to.
So this is what?
So you'd normally like visualize your sample. You have all these focus knobs here. Okay, so of course you'd have to use these knobs to get into focus. Okay, so you have the coarse knob and the fine knob. You have these two buttons here. One would bring the objective down, which is actually the same as this one. So it brings the objective down, and then the other one would bring it back into focus. Okay, you can increase the amount of light that you have instead of using the exposure time. You can increase the amount of light by putting that up, or using these arrows. So 19 right now, or you can also use this here. Just increase the amount of light that's getting in. Okay. So, first step of laser capture microdissection would be to find out the cutting functions. What type of function would you use to cut your sample? So you have, if you click here, down here, you have all these different cutting functions. Okay, so we have a cut here, then you have joint cut, close cut. These are all for just cutting your sample without collecting anything at all. Okay, then you have LPC. This is laser pressure catapult. That's what LPC means. So it just catapults one dot, like a dot, a piece of a very small piece of your tissue. Then you also have this line auto LPC, the close cut auto LPC, auto LPC. As long as it says auto LPC, that's for just ordinary glass slides. So it is possible to put your tissue on just an ordinary glass slide and cut it. I will show you later what it does. And then you have this Robo LPC, the center Robo LPC. That is what you use for collecting tissue. So you cut large pieces of your tissue, or you cut the whole cell itself, and then you can use the laser to lift that cut tissue into your collecting vessel. Okay, so to demonstrate the different cutting functions, you basically have to bring your membrane slide on an area where there's really nothing except membrane. Okay, so no tissue yet. All right. So you can move the stage using different. You can move the stage in different ways. One would be just to use all these arrows on your keypad. So you just basically, yep. Moving it.
Well, this is like the second one.
Okay.
I don't know if you remember it.
Let's see. Just have to see over focus. Okay. We can use this other one here for moving. Okay. There is a speed attached to the movement of your stage. So this one is for scrolling. So once that arrow appears, you bring your mouse to the sides. It can be up or on the sides. Okay. That speed would be at 15 right now. So if you want to make it faster or slower, then you can just adjust this. There are other speeds here. So if you go to the arrow keys, so it's at 20. So okay, it's moving. So you use these arrow keys here to like just move your stage. Just find an area which is empty, or you can find where your tissue is located. Okay. Another way to move the stage, of course, would be using the joystick that's right here. Okay, so you can use this to move your stage as well. One other way would be to use stage mode. So stage mode is, it's basically freezing the stage, whatever is there. So you can use your mouse, basically you just move your mouse to move the stage. So you have to hit this F7
So those are like little.
and then use your mouse to move the stage. It's a little bit more difficult to do it this way, but there is like a speed also attached to that. Okay, and once you want to get out of stage mode, you have to click on this F7 again. Yeah. Okay. One other way to move your stage would be to use this navigator. So click on N. So you have 3 slides here because we are using a specimen holder that can hold all 3 slides. So this arrow here and whatever is highlighted here, that's where you are right now. So you are in this position. So if you click somewhere else, that moves the stage faster. So it's a faster way of moving. And you can go from one slide to the other by just double clicking. Okay, there. Right. Okay, so that's the navigator button there. Of course, every time you move, the focus is going to change, so you always have to make sure that you are in focus. Okay.
So I'm a little bit worried about the.
Alright, so let's just focus.
At some point, if you're gonna do
You can also change from one objective to the other. Let's close that. And let's go to 20x.
You can see how dim the
Wow,
that's here.
Oh.
Right. And of course you have to make small adjustments in your focus as well. You can go to the 40x.
Or it could be multiple changes or something.
So it could be
Sometimes,
pretty
you know,
good.
the maybe the humidity chambers, if they were well
Comes
comes
in
in contact with the
Normally it would take slower
But you don't know.
to see where you are, just because your exposure time is a little bit more.
Yeah, this is.
I'll just take 5 for
Okay.
this number slip.
Alright, so let's demonstrate the different cutting functions. Let's go back to 5x.
Oh, wait. This is our negative control.
Okay, so
It's not negative.
we start with cut. So usually if it's in this LPC mode, it just is stuck there. So what you do is to just click on anywhere, then you'll be able to go to the normal. Okay, so we cut first, and then so you choose your cutting function, which is a cut, and then here under this would be where you're collecting your tissue. So if it says manual, then you're not going to collect anything. So there's no catapulting of your tissue. Okay. Then there's also, whatever, depending on what collecting device you're using, then you choose that. Okay, for now, let's just choose manual because cutting doesn't really collect anything. Okay, we have all these drawing tools here. Okay, so you can choose here, you can choose this wavy line, or you can just choose a line, or you can do a square, triangle, or rectangle, or a circle. So you choose a cutting function, and then you can just draw
You could resolve these big
whatever
ones better.
region you're collecting. So this is just to demonstrate a cut. So for a cut, whatever you draw has to be closed. Okay? Or, I mean, if you don't close it, then it's not going to cut the whole thing. It's just gonna cut wherever you drew it. Okay? So once you have this, you can activate your laser by hitting this button here. Oops. Oops. Actually, I forgot to demonstrate. We can click on
See
stop.
how much smaller they actually are,
Okay.
right? I mean, 'cause that measure the brightness or the size.
it remembered because we drew we had some elements drawn earlier.
Probably more molecules.
So
Probably way more molecules.
I guess we can start here.
And this one, which is the biggest, which actually does look.
So let's just go and go back to this. This is called an element list. Okay, so this shows you what were drawn. So in this particular slide, we are on slide 3. We drew this
It's more.
earlier. You can go here and click on motion and say go to element. Okay, and that brings your element to the middle.
If you wanted to know
So let's
how much
just click anywhere here. Okay, we didn't really draw anything here, so you can actually delete that. So you can just there. This is for deleting the last
Yeah, but you could say if this
figure drawn. I don't know what was the last one. I guess never mind.
Is there a way to measure?
So what you do is just to draw something else again, and then we have to highlight it so that the laser knows that
all
that's
those
what we
pixels have the
want
same value.
to cut.
So you just have to take pictures like this if you actually want to.
Okay, once you've drawn that, there's this arrow here. Click on that and click on this. Again, there has to be a box drawn on it.
We'll get done what we can.
There. So once it has the box, then the laser would know, okay, this is what I need to cut. So you then activate the laser. It brings whatever you want to cut to the center, and it starts cutting. Notice that it just cut it. It didn't lift the tissue or anything. Okay, so that's a cut. Let's go to the other tool here. This is the joint cut. Okay, and then you can just draw something.
And it also helps sometimes, you know, if you can see what you're actually pressuring.
So for a joint cut, it doesn't really show it here. Can you draw a bigger one?
Decide, you know, at what point you're going to set your threshold.
Yeah.
And it's a little tricky, but you come up with something. So this one's good and it's really all this one, but not this one.
Okay, so for a joint cut, it's supposed to show a bridge in between. So if you go here, okay, this one is for adjusting the distance between the joints. Okay, so for a 5x, let's just increase this for now. So let's make it 30. It's just to show.
Just give you one region.
Okay. Okay. So notice that there's a space that is drawn even if you close it down. So if it's a joint cut, there's still an empty area here which will not be cut depending on how much laser you have. So if you highlight this again,
Like if I made it this long, could I compare this one to this one?
okay, and cut
Or
it.
is
Well,
it
this
this one
one-
is out of the threshold?
This one with this threshold, yeah, it's I'm not, you know, we have to think a little bit about what's the best way to do this. You set the threshold for both of these.
So it would stop cutting. Okay, so
You're
that's a
gonna
joint
measure all
cut.
the
Then the
things.
next one here
So,
would be a close cut.
but then, so this
So,
one isn't gonna work because
Draw something where you don't close
this
it
one's
up.
only good for one region. Like, this is a simple one. This is called what was region statistics. So region measurements.
Okay, so notice that it cuts. I mean, it closes the circle, and then you can just go ahead and cut it again. Okay, the next one here
Getting everything in a region.
is called LPC.
This is
Okay, so that's where you have just a dot.
Well, what you can do.
Okay, and then you can just click somewhere.
Threshold.
Okay, and then wherever that dot is, that's where the laser would hit. So if you click there,
Create region drawn objects, and it creates a region around every object section.
it actually, oh, it again,
And
it
then you
remembered.
can go to
It remembered again the last one you drew.
measurements.
Okay, so that's the close cut.
And you can configure for what data you want to collect.
And then just click on OK. That message will show if you don't have a collecting device, because LPC is a mode where it also catapults the tissue. So notice that it bored a hole there, and that whatever tissue was in there should have also gone up. Okay. So for the next laser functions, again, if it goes here, click on somewhere else. Okay, there. The next modes would be for catapulting already. So you would have to have a cap in here. So you choose one of these depending on how many adhesive caps you want to use in your experiment or whatever. So you can use this, put the cap. So you can choose your cap. Let's just do the adhesive, the clear one for now. Okay. So to put this on, the cap adhesive side should be facing down because it has to be facing the tissue. Okay. And there's this slot here where your cap would go in. Then there's a place for it to tuck in your tissue. So you have this, right? Then you put this on.
Oh yeah, I guess that is. It's usually that one. We usually go into.
And then you tuck it in this way.
Is this toolbar?
And then you have to make sure that your cap is flat.
And if I open it on my computer, like later,
Can you put it on?
Well, you would have to open it with the Metamark soft.
Yeah, no, that's what I mean.
Yeah.
Okay, but I could.
Then you could do all of this.
Okay.
Then you could do all of this. But you would have to decide if you like where that threshold is. You might want to set separate thresholds. Places. There is another tool that will allow you to look at local differences in contrast.
So there. And this is magnetized here. So there's this cap mover here, this slot here. You basically just put this on that way. Okay.
So
now you have to make sure your cap would go a certain distance, so it goes over whatever ROI you drew. Okay. So if you choose your line auto LPC, that's basically for just a line. Okay. And it's for just ordinary glass slides, but I'll show you how it works. So you select a line tool and just draw a line. Then you notice that there are all these dots here, and the spacing between dots can again be adjusted using this function here. Okay, so that's the distance of auto LPC shots. So you can make that farther apart or closer by just changing the number right there. Okay. So if you choose this function, and you want whatever you collected to get into your cap, then basically you have to tell the cap to go there, right? So let's go to this one. Oh no, that's for this one here. This shows the collection device dialog. Okay. So you have to make sure that whatever is displayed here is what you're using. So this says single cap. That's not the one that we're using. So you have to change the collector and go to the single tube collector 500. So this is a 500.
What you can do is you can make an Excel
click on OK.
file and do that. Or we have another way of doing this where you can actually put it
Okay, so it's displayed now. You want to make sure it goes down, because once it travels, it has to go down a certain distance. Okay, but you have to make sure that it's not going to touch your stage. So you have to go to this adjust. There are two movements. One is the moving height, and the second is the working height. Moving height is how low it would go to travel. So you can click on this go to just to see how far it goes. Okay, and as long as it's not touching your stage, then you're fine. You're fine with it. Okay, you can just go to that, or you can just put it up or down by clicking on the arrows there. So if you try to bring it up, you can see that it's going to go up a little bit. Okay, then once you're happy with your adjustment, you have to set that. So you click on set here so that it remembers that it's what you we have this already drawn. So you click on the tube, make sure it's the tube that you hit. So it goes down and goes right on top of, so it's there already. Okay? And since we're using a clear cap, then you easily see it. If you're using the opaque cap, you'd have to adjust the gain somehow because it's opaque.
But sometimes by looking at the data, it's brighter.
The next thing to do would be to make sure that the distance, which I think it's too high, because I think we put the cap on wrong. Okay, so your distance between the cap and the tissue should be about 1 millimeter only. Okay, and it's not, and it's this metal part is already close to your cover slip, so you have to bring it back up by going home. Here. Okay, take this off. And you have to make sure that this cap part is a little bit lower. So this has to be lower. Okay?
Yeah.
If it's really tight, then that's the wrong. There. You have to put it like lower.
Is it, am I on?
Here.
Is there a way I can check?
You ever get?
Okay, so your cap is lower now. Then put it back here.
I've been on it for
Then you go ahead and click on the cap again.
Yeah, I don't know. I could try and look it up.
Okay.
I think we put all the users on it.
Now it's lower. Okay?
Maybe I just
So, you have to make sure your distance is about 1 millimeter. Again, we're using this one here, the working height. That's the
This is
working
all
height.
nice.
So,
whenever
for
you do
adjustments,
line,
use these arrows here. You are there already. Okay? So, let's try going up a little bit. And notice that it's going up. Okay? But if you're not, if you're going too far out, then and you want to go back to your original, then you don't really need to set. You just go to
means it's going to open
whatever was set earlier. Okay? And then you can close that.
Data.
And then let's highlight this again. Okay? And let's start the laser.
Complicated if you do two different things in every.
So that's how the auto LPC works. It doesn't cut through a large region of tissue. It only catapults whatever is in these dots here. Okay? And it's very effective for glass slides. Next one
So there's lots of ways to do this.
would be the auto LPC. Okay, so this is for bigger areas. Now you can draw a shape.
But then you may want to take.
And again, all these dots can be adjusted and go hit your.
Because the integrated morphology doesn't ever like the reset.
So it's like a gun.
But,
Here.
yeah. But
Next one would be the close cut and the auto
No, you have one
LPC.
like this. You can
So
always.
this one would cut
But you can't
along the region that you drew, and at the same time do all this catapulting. So again, let's draw.
Oh, this is in
That.
So I know you can. Is this? I know there's the whole localization thing for super resolution, but would I be able to
So that's
see
the difference.
if a single molecule of bromine was not surrounded by any others? Would I be able to see a spot like this? I mean, it might be like a dim
Okay.
spot. But would that
Next
be
one.
possible?
Sure.
This is for membrane slides, the Robo LPC.
I guess
Okay.
theoretically.
So this is what you use to cut whole sections of tissue.
You'll be able to see, pretty small things. I don't know how big this is.
Okay, and notice when you draw, there's this dot here. So there's a space in between and a dot. So this is where, so your laser would cut through here and then lift your tissue at this point.
See that? And
Okay.
if you knew that they were separate, you could say, okay, those are individual leaves, but I don't need to know how many molecules. I don't know.
Yeah.
There's probably still several.
Okay.
So,
Because at most, I think my chains are labeled with one molecule protein.
Okay, now
So
notice that the tissue is not lifted. So there is something that you need to adjust. There is this cut energy here, and there is this delta. Okay,
yeah, I don't know.
the sum of this cut energy and delta
yeah.
would be your
Unless you
laser
absolutely know you have
pressure
some
catapult energy,
you
the
can put
LPC
on
energy.
And look for
So
it.
the maximum
But
should
how do
be
you know?
100,
How do you ever know?
100.
That was Becky.
Okay, so if it does not lift the tissue, it's either your cutting energy is too low or the delta is too low. Okay, so you'd have to play around with this until you get, the tissue off from the membrane. Okay, so let's say we increase the delta to 45. Okay, and we can try doing another. So before you even collect your own sample, you have to make sure that you have optimized all the settings of your system.
We'll probably catch up because I'm only going to shoot like 5 or a couple images of the
So still, it might be also possible that the distance here is too much. Okay, so let's try adjusting this and bring it down to 20 again. Just click on OK. Maybe we can try increasing the laser power a little bit. Make it 48 maybe. Okay, and again, let's draw something. Let's adjust your focus.
It was just a curiosity because I have a lot of separated spots. I was just curious if it could be like a single. We have chain polymer.
I want to do that. I want to hit split. So I'm not going to say that one.
Are you sure your slides are not inverted?
Let's do the laser.
Yeah,
Go
we're
to
in
60.
normal, but it should
60.
still-
The laser had to be 63.
63.
60, yeah.
Okay,
to
so
be
let's
able
do
to cut.
this, make it a tube. Okay, so that we'll be able to collect something
Yes.
there. And then you have to make sure that whatever you draw will really go to the tube by clicking on the element. So let's draw something first. Okay, and then let's go to this element list. So this is number 23, Robo LPC, and it's going to the tube. Okay, but let's play around with the laser power still.
Make it to 63.
Increase that.
Uh,
63.
And then we have to decrease that then. And then let's decrease that.
Oh, I see.
57. Okay, and let's cut.
Yeah. Like, so you come up with like, like histogram that's like, see, I forget. I see, so if I have like the base one. I see, okay.
Notice that if you cut with a higher laser power, the thickness increases. Again, still,
Still not
it's not
cutting.
catapulting and not sure what's wrong.
And we're just using Ruby. And I don't know, 'cause I know they have the photo, they have the photo stretches.
Maybe let's try the
It's
tissue.
also possible that
It's wet, moisture.
it's
Let's
either wet
try-
or it's inverted. It's
Yeah.
not in the right.
This is the slides. Let's try the tissues we highlighted.
Okay, so there is an ability here to remember areas that you have chosen earlier. Okay, so let's go to motion.
So we,
Okay, let's see. Maybe.
there's
How was that?
I found sort of a reference that
Okay, oh, let's load this. So look, file, open elements. Let's go to slide 1
Yeah.
and then okay. Let's say elements. No. Okay. Did it go? Okay, so that's. So we are here in the slide. So you go to slide 1, highlight slide 1, and then here, then motion, go to element.
Our backgrounds.
Okay. So earlier we used this flag.
Well,
Here.
we don't know if there's actually matrix.
Just clicked somewhere close to the tissue, and your system will be able to remember that. Okay, so you can put as many flags as you want there. Okay, so let's try
save us a little bit of time.
doing the RoboLPC on this.
All right, now we're gonna.
Maybe just on the membrane first.
But usually what people do, like any else is which is you turn it on but you just sort of the more you do, just start.
I will look at the negative.
It cuts it, but it doesn't catapult it. I'm not sure why.
Do it again.
Your tissue might be. Let's go. Let's go 60 and make that 40.
It's on the web. It's like 488.
No, last one was a negative.
And then let's close down
That's why
the.
it's just like,
Make it 15.
still not a lot of background.
Okay.
So it seems like
Let's cut again. Something
Why
else.
not at like a low level all the time, and then it gets upregulated.
Yeah, she's right. It's
Yes. But so it would be interesting to look at the brightness between the lead and
there's 405, 488, 561, and 6.
Okay, so there's a little bit of lift, and now it's done.
Beautiful.
Okay. So 60/40. It seems okay. So that's a RoboLPC, and then there's the other one. The last function there would be the center RoboLPC. So when you draw your ROI,
All this stuff, like, I'm not really that.
instead of the dot being at the side, it's going to be at the center.
plenty of cells down here. I'm just gonna try to find an area where they're not.
Okay, so basically it would catapult your tissue from the center.
No OPS, but Y27. Okay, let's try this area.
Yep. Okay, now this time that's not gonna work because there's still an area that's so that might be again something to do with the distance between the joint, the distance of the joint. So it's still holding the tissue. That means it's a little bit too big still. So you probably would still have to like decrease that joint. Yeah, make that maybe 12 or something. And then just click on OK. Okay. Anyway, so there's also another way to, like, if something is lifted already that way, there's another way to lift it up into your vessel, and that's by clicking on this one here. Okay, so if you hover your mouse over here, it tells you, it catapults whatever position you clicked into your vessel. So if you click somewhere here, I don't know if it's gonna do it, but yeah. Okay, so it will still bring it up. Alright. Okay, so once you have adjusted your cutting parameters, energy-wise, it probably would need more energy to cut through the tissue itself, because you're not just cutting the membrane but the tissue also, so you'd probably need higher energy, but we'll see. So you can choose a part of your tissue and, you know, draw a region there where you want to cut. Make sure that you adjust your focus as well.
Battery almost.
And then note that what you drew here is using the center robo LPC. Okay, so if you don't want to use that, after you have drawn this, you can just go back to your elements again. Okay, then highlight that, and then this one here, you can change that to the robo LPC. Okay, and then make sure it's going to your tube. Okay, and then now we can just there. Okay, so there you have your tissue collected. Now if you want to be able to see what you have collected, you can use this
Yeah, there's
cap check. Let's cap check. Go ahead.
So I might be able to tell more.
So your cap is going to go, your stage moves towards this hole here, so that your objective goes over the, your cap goes over the objectives. Then you will have to adjust the focus, okay? So you go, bring your objective higher, so until you see something. And it's usually always good to just go with the 5x objective, so you can see a bigger area. And notice that you see all this because we used the auto LPC function earlier. And there's all this small there, that's the one that you collected, right?
Way to look at the bright side.
So that's a way to check
That's true.
what you
I
have
do
collected,
hate that. I'm like,
right? Then let's just bring it to the center again. Use the arrow keys or bring this to the center. The other side. Right there. Okay? See. Okay? Now, if you're done checking, then you can go back to your to your slide by clicking on this cap check again. This time it has an X. So it moves back to the last position you were in. And the focus, it remembers. Okay? And it also remembers the objective that you used. So if you were using a 20 or a 40x to cut, and then you go to the cap check with a 5x, basically when it goes back to the slide, the objective would go back to whatever objective you used to cut the system. Alright. What else? Let's
Can
just
you show
change this.
the save pre-cut, after cut pictures?
Oh, okay. So, if you go to settings, we can actually save whatever pictures you set it to save. So if you go to preferences, okay, this one here under configuration can save your settings file. So if ever you like change the energies on your whatever sample you have, then you can actually save that setting, and then when you come back you can use those same settings by saving them here. Okay. If you go to elements, you can also save whatever is in the element list. Okay. So that if you want some information on maybe that how much or how big of an area you cut, which laser tool you used, which objective you used, then you can also see it in this element list. For images, you can save images by putting in a base name. Okay, so let's say the name has this base name, then you can put it in a folder. Right now, I think it's going somewhere else. So if you go here and go to your folder, where is it? Ted's folder? Or
I think you learned how to do a lot this summer. And
Yeah, I think
things are in the lab. And
Yeah, definitely. And it's good for the year too, because it's like less of like having to just like make sure everything is on
maybe here. And then let's see if you go. So we can make a new folder under this if you want, or it might be already here. So just go down. And
The cells aren't really like,
they're kind of in between.
yeah, there's like in between.
maybe it's under my document.
It's under TED.
Okay. Oh, it's under TED. Okay, so it's under TED then.
You go down to TED's lab.
The changes that I'm seeing are in areas that we know.
So you save wherever you want to.
Users directly to you under user.
Okay. Oops.
Not under my
There.
documents.
So this you find it somewhere here. So TED. That's L. So you go T. Go to T.
Yeah, okay.
TED lab.
Let's lab. Go down. Today demo video.
Okay.
Yes.
And then just click OK. Alright, so all the images that you want to save will go here. Okay, you can just put a file name. Okay, then under laser, this is where you choose whether you want to like save an auto image. Okay, so it's either you choose the ones that you cut before and after, or since you're doing an LPC, a laser pressure catapult, then you have to choose one before LPC and after LPC. Okay, then you can choose whatever format the picture will be in, so you can save it as a TIFF, as a bitmap, or a JPEG. Okay, so right now it's a TIFF, and then just click on OK. So I don't know if those images they probably were already saved.
Yeah.
Somebody else's folder. So I'll have to get into that later on. But anyway, let's draw another so that we can see the images before and after. It's okay, we are here. Yeah, so you just change that also, make sure so that the next ones would be on Robo LPC. Then just go back to elements, change that. Okay.
Okay. Sounds good. I'll be back up in 15 minutes or so. Thank you for your help.
Again. Okay. And if you want to view whatever was saved, you can go to this Explorer here and go to your computer and system, users, and all that, and then you go to your demo, and then this. So this is after, and then you have the before image. So go ahead. So that's the before, and then this after. Okay? It's like it cut it. Yeah, and then you have all those images already saved in the folder. Okay, so we can close that. Okay, what else? This is also, I'd like also to show you what the energy does in terms of the cutting line that you have. Okay, so let's go again to an empty area. Okay, and then just draw some. Okay, right. So notice that your energy is 60. Okay, so what you need to do, let's change this to just a cut. So go back to elements. So if you drew the ROI first before changing, then you have to always go to the element list to change it from there. Otherwise, if you do it here first and then draw your ROI, then it remembers what you did. Okay, so what we're gonna show now is how your laser energy will affect the cutting line. Okay, so basically if you activate your laser and then just go through here, make it maybe higher still. Okay, so just use the arrow keys and increase that. We'll see how the cut. Okay, go ahead, increase more. Yep, more. It's higher and higher. Okay, so the higher the energy you use, the thicker the cutting line would be, and the rougher it would be. Okay, so you always want a line that's like smooth, not too thick. Okay, depending on the distance between whatever you're cutting. Yes, there. Right. So you can like remove this last element so that you can see it clearly. Oh, this one too. Let's highlight it, and then next. No, that one. Just delete. First we delete here. There. Okay. So thickness increases. The delta also, of course, will tell you how much laser is used to lift or catapult the tissue onto your collecting vessel. So if it's too low, then obviously it's not going to go up. If it's too high, then it might go somewhere else. Okay, it still would probably go into your cap, but you have to always be aware that your total here should be 100. Okay, so right now it's over that, but we're not doing the LPC, so it's okay. Right? But once you get to the LPC, it has to be just 100. And you can actually see it from here. If you uncheck this, auto change, calculus delta equals LPC, then you can see here, this is your LPC energy, which should include your delta if you're doing your Robo LPC. Okay, let's go there again. Okay, so you check that so that you can manipulate the delta value. See, it became 0 now. because you were at 100 already. So let's just go back to the 60-40 earlier. So instead of going here, you can also, cut using that. And then if you want to stop whatever cutting you have, then you can just click on the stop. Okay. We can also move to the next slide. Basically, we have another flag there. So if we bring your collecting vessel home, you just go back to the collection device dialog and go to operation and click this home till it goes back. Okay, then you can go back to your element list, and then I think we saved it in slide 2. Okay, so if you click on 6 and motion, go to element, so it moves to, okay, just click anywhere else, and that's another flag that we did earlier, which is just an empty part of the membrane. If you can go somewhere else, that's the tissue there. Okay, so we can collect, different areas of your tissue. You can draw regions of interest maybe at the edges, or you can collect somewhere in the middle, or you can collect the whole thing, okay, depending on what you want to collect. So
Try one layer first. Try one layer. Draw focus first. Yeah. Try cut a hole.
okay, so it's a Robo LPC and a tube, so it remembers that. So if you commence the laser, start it, so your tube would already go on top because it knows that it's supposed to cut and catapult on the tube. Oops, of course it remembered that other one. I think it's remembering all those flags. Okay. Okay, so there's still some part of the tissue that's left there. You can use that. Okay, there. Right. And then you can draw as many regions as you want, and then hit the laser so that it would do them one at a time. So go ahead. And you can also do the drawing on, other areas as well, and it will remember that. Okay. Then that's probably a thicker part of your tissue. And that's it fell away there. Too much. other tissue, maybe we can just do another cut there. Oops, now it flew away. Okay.
Okay, let's go over, shut down the instrument.
Okay.
Yes.
So, once you're done doing all your collections, you have to bring back your collecting device home. Okay. Then you have to take out this. You have to have some buffer ready. Lysis buffer, for instance, or whatever buffer you need for depending on whatever your downstream analysis is. You put your buffer on the tube and then you close it down and you have to store this inverted, okay, so that there's contact between your buffer and the tissue. And then you can like put this on ice and bring it to your lab. Next thing to do is to centrifuge this so that you'll be able to have your tissue mix in with your lysis buffer. And then once you have it here, then you can bring it over for RNA, DNA analysis, and for protein analysis. There you go. Then you have to take off your slides, okay? You can take off this whole thing if you want. Okay, so you can bring your slides over there, and then to close down the system, let's just turn off the light. The light, by the way, is on the whole time. Okay, so you can choose to save all of these elements or not. You can delete them all if you want. You can just click on X and that deletes everything. Okay, you can close everything there. You can close that. Go. Then if you have some pictures, then you can transfer them from the computer to whatever your device is. And then for this part of the setup, you can just log off, and then you can just close all of these by just using so we have this here. Those are just the two switches which control everything. So everything's turned off from there. Okay? Any questions?
That's really good.
This is not everything yet, because there's still the part where if you draw the ROI and it's not right there, yeah, it's the calibration part. Those are all other things that.
Okay. So we'll do another session?
Yeah, maybe. It's
Yeah.
up to you.
Yeah, because we do another session.
Yeah.
I'll go back and see how this thing works.
Yeah.
And then we'll